Hello, everyone. Welcome to my video tutorials for spatial data analysis. Today, I'm going to show you how to analyze the Xenium FFPE human breast cancer dataset using the Squidpy package. So let's note the packages for today's analysis. If you want to use this dataset for practice, you can download the dataset from 10 times Genomics website. We can go to the 10 times Genomics website and have a look at this dataset. You can see this is the 10 times Genomics website. They made this dataset publicly available to demonstrate their technology. And also they published a paper for this dataset. If you want to know the details analysis, you can read this paper. So here is the information for this dataset. You can see they did in situ hybridization for the human breast cancer tissue. And also they performed the immunohistochemistry for the CD20 and the B2 antibody, and also stained the tissue with DAPI to reveal the cell nuclei. So here is the tissue information. They also analyzed and annotated the cell types. But for today's analysis, you need to download the data from the output files. So you can see they put all the files in one zipped file. If you click this link, you will download all the data. I downloaded the data already. We can have a look at the data in my folder. You can see here is the data that I unzipped from the downloaded file. You can see we have the cell feature matrix data. This is the barcodes, features, and the matrix that we normally use for single-cell RNA sequencing data analysis. And it also has the cell feature matrix data in H5 format. And here is the cell metadata. You need to unzip this cell CSV file as the cells. Then for today's analysis using the Squidpy, we only need the cell feature matrix H5 and also the metadata in the cells CSV file. So now we can load the data into Python for analysis. So we loaded the packages already. First, we can use the read 10 times H5 function to load the feature matrix data as A data. Let's load the data. We can have a look at the A data. Let's run. You can see at the moment we have 167,780 cells and 313 genes. You can see at the moment we only have the data variables, gene IDs, feature types, and the genome. We don't have the data observations and the spatial information. So we can load the metadata. I showed you the metadata is in the cells CSV file, so we can load the metadata. Then we can copy the metadata from the cells CSV file into the data object. Let's run. If we have a look at the data now, you can see we add the data observations. Now we have the cell ID X and Y centroid, transcripts count, control probes count, control covert counts, total counts, cell area and nuclear area. Because we are analyzing the spatial dataset, we also need to add the spatial layer into the data object. We know the cell coordinates are in the metadata X and Y centroid, so we can get the X and Y centroid information and add into the data object as the spatial layer. Let's run. If we have a look at the data now, you can see we added the new layer spatial into the data object. So now we can calculate the quality control matrix to perform quality control for this dataset. We can use the calculate QC matrix function to calculate the quality control matrix. Because the in situ hybridization also perform the control probes and the control covert analysis, so we can also calculate the percentage of control probes and the control code words. So let's calculate the percentage of control probes first. You can see the negative DNA probe count is about 0.08%. Next, we can calculate the percentage of control coverts. You can see the negative decoding count is very low according to the percentage. So we calculated the quality control matrix. Now we can make a histoplot to show the distribution of different Q metrics. We can plot the total count, n genes by count, cell area, and also the nuclear ratio at the same time. The nuclear ratio will be calculated by the nuclear area divided by the total cell area. So let's make the plot to see the distribution of quality control matrix. You can see we generated 4 histoplot here. The first one is the total count in each cell. You can see the x-axis is the total count in each cell, and the y-axis is the total cell numbers that have the same count numbers. You can see majority of cells have the count under 500, and the highest count is around 1250. You can see the second histoplot is the unique transcripts per cell. Again, you can see majority of cells have the count between 0 to 150. The highest count is below 200. Then the third histoplot is the area of segmented cells, and the last one is the nuclear ratio. So we can set a threshold according to the histoplot for the quality control matrix for filtration. So we are going to set the minimum count per cell as 10. Then for the genes, we only keep genes that have been detected at least in 5 cells. So let's filter the data using the threshold we set here. After data filtration, we can have a look at the data now. I put the gene numbers and the cell numbers here. Before the filtration, let's filter the data now. You can see now we only have 164,000 cells, but we still have the same number of genes. So after the filtration, now we can perform the standard Squidpy workflow to connect the cells. Because we only have 313 genes here, so we are not going to perform the step for identify the highly variable genes. We only normalize the data, then log transformation. After that, we directly perform PCA analysis. Then we identify the neighbors, run UMAP, and use the Nathan method to cluster the cells. So let's run the standard Squidpy workflow to cluster the cells. We can run. Okay, we finished the standard workflow analysis. Now we can visualize the cell types on UMAP. Let's plot the cell clusters according to their Nathan cluster numbers. You can see we use the resolution at 0.3 in the standard workflow analysis. Then we have 9 cell clusters here. We can use some mark genes as example to show you how to identify the cell clusters. For example, we can use the ITGAX. It is a mark gene for macrophages. VWF is a mark gene for the endothelial cells. Then FASN is the mark gene for tumor cells. So let's see the expression level of those mark genes in different cell clusters. You can see we have the feature plot. You see cluster 3 is macrophages, cluster 6 is endothelial cells, then cluster 0, 4, and 5 are tumor cells. If you don't know the marker genes very well, you can watch my Squidpy video tutorials to identify differentially expressed genes for each cell clusters. Then you can use those marker genes to annotate the cell clusters. So in this video tutorial, I'm not going to show you how to identify marker genes and how to annotate the cell clusters. So we cluster the cell types according to their gene expression. We have 9 cell clusters here. Because this is spatial data analysis, now we can visualize the cell clusters on spatial coordinators. We can count the cell clusters as Nathan clusters number, and at the same time, we also show the gene expression for FASN because it is highly expressed in the tumor cells. Let's run this demonstration. Okay, you can see from the figure down here, the first one is the Nathan cell clusters from 0 to 8, 9 cell clusters on the spatial coordinators. And on the right-hand side, you can see FASN expression in the tumor cells. So according to the cell cluster color, cluster 0 in blue, cluster 4 in purple, and cluster 5 in brown, they are tumor cells express high-level FASN gene. Okay, that's it for today's demonstration. I showed you how to analyze the Xenium human breast cancer dataset using Squidpy package. We cluster the cell types and also we can visualize cell clusters and gene expression on spatial coordinators. Now you can use this data object to perform more analysis. I hope this video tutorial could help your data analysis. Please subscribe my channel if you haven't to do so, and share my videos with your friends. Thank you.